Kia ora koutou, ko Ange tōku ingoa. Welcome to our Growing Point workshop at Dignan Street Community Gardens of how to measure Brix levels as an indicator of plant health and how to increase that plant health with foliar feeding. This workshop is sponsored by the Auckland City Council Waste Minimisation Innovation Fund. Thanks so much for coming out on this cold for Auckland autumn day. This morning we're going to be talking about measuring Brix levels. Brix is a measure of the total soluble solids in plant sap. It measures the percentage of sugar, vitamins, minerals, amino acids, proteins, hormones, and other solids. The more a plant photosynthesizes, the more Brix they will have. So levels will be lower in the morning and grow over the day as the sun shines and the plant photosynthesizes. They expel some of the sugars into the soil to feed the microbes, who reciprocate by breaking down minerals into soluble forms that the plants can use. So the equipment we need for this is your refractometer kit, the pasta pipette for dropping, a garlic crusher to squeeze the juice out of the leaf, and something to clean it with. Brix is an indicator of plant health and nutrient density. As the Brix comes up, we see an increase in biological activity, mineral availability, and plants become resistant to frost, pests, and disease. We also see deep soil carbon sequestration, water, and nutrient cycling. Plants with a low Brix, reading less than 2, have one root in the grave. They're at the end of their lives and will soon be consumed by insects. They rot quickly. At a mid-level Brix, from about 3 to 7, plants will be substantially healthier. At the higher end, around 6, plants are thriving and tastier. Insects cannot tolerate a level of Brix of 8 or more. They do not possess a pancreas, so cannot process the sugars, which are toxic to them. Plants will be thriving, are very nutritious, and taste a lot better. A sharp line on the refractometer indicates low calcium levels. A fuzzy line indicates good calcium levels. Just try and get some of the liquid out. There's some there.
It's really fuzzy.
Oh, what does that tell us?
Calcium.
High calcium.
Oh.
Means there's lots of
fuzzy is
calcium.
good.
Fuzzy is good. Hard to read, but yeah.
Using a refractometer, measure Brix sap in the afternoon and then again in the morning. The level should drop overnight if sufficient boron is present.
Oh, we've talked about about measuring the Brix levels in the plant by using the refractometer, and now we're going to talk about how to increase those Brix. So the plant has just as it has a microbiome in its rhizosphere, it also has a microbiome on its phyllosphere, which is the surface of the leaf. So that contains beneficial microbes, just as we have beneficial microbes in the soil, like bacteria and protozoa and bacterial feeding nematodes. We also have a whole lot of organisms on the leaf, and these do a really good job of increasing plant Brix by providing immunity to the plant. So the plant doesn't have its own antibody system or immunity like we do, and so it really relies on on the microbiome on its leaves to give it that immunity. We want to get the spray as fine as we can. So the spray we're using today is made by flushing water through worm farm and catching it fresh. If you do it like that, you'll get the beneficial aerobic microbes